I'm Sarah Berry. I'm a nutritional scientist at King's College London, and I also work with ZOE, who are a startup tech company. I need to make the disclaimer at the beginning that I'm not a data scientist, I am a nutritional scientist, so I'm going to be throwing some nutrition into the talk along the way, and please also excuse any incorrect AI terminologies that I may make. So what I'm going to do first is talk about how AI has enhanced my experience as a nutritional scientist and how it's really allowed us to press the fast forward button on our research. And I'm going to do this by discussing our PREDICT program of research, where we've successfully combined biological science with data science. So the PREDICT program is an ongoing research program to measure responses to food. It's a collaboration between a number of academic institutions led by King's College London and ZOE, this startup tech company, who was specifically formed for this purpose. And uniquely, it combines traditional nutritional academia with a tech company of data scientists, machine learning experts that really allow us to achieve this high precision and high quality output at a scale and a really unprecedented pace. But why am I as a nutritional researcher, and why is ZOE as a tech company doing this? Well, diet plays a huge part in disease and is accountable for about 11 million deaths a year and about 255 million disability adjusted life years. So it plays a huge burden. And despite the global and national nutrition recommendations, the advice just obviously isn't working. We believe this is in part due to the need for more personalised nutrition advice. And we need personalised nutritional advice because we're complicated and food is complicated. And nutritional research is only really just starting to scratch the surface of understanding how food affects our health. And this is because food composition itself is complex with about 26,000 chemicals and interacting chemical pathways. But also the form in which we eat our foods is really complex. So for example, a whole apple or a pureed apple have exactly the same nutrients, but will have hugely different effects on our body. And to make matters even more complicated, we're incredibly complicated with our millions of biological pathways which differ between every individual. So we all respond differently to food, and what's right for me might not be right what's right for you as individuals. But nutritional advice is based on population averages, and the guidelines, as we know, takes a one-size-fits-all approach. And also nutritional studies typically mean response. So the blue line here from one of my studies shows the increase in blood fat over 8 hours after consuming a bag of nuts. And this is what was reported in the published paper. But actually, each individual's response, which is shown here by the individual lines, was hugely variable. And this is despite being in a really tightly controlled clinical setting. So how relevant is the mean, this blue line, for the person that's up here or the person that's down here, which really raises the question to us of how many of us are average? And I think it really shows the place for personalised nutrition. But if we overlay the complexity of us and the complexity of food, understanding how foods impact the health of each and every one of us really requires a different approach to the one that we've traditionally taken in nutritional research. So in the past, in nutritional research, we've either relied on very low precision but large scale data using rather imprecise methodology, or we've relied on very small scale but higher precision, higher resolution data from randomized controlled trials, for example, using precise but very burdensome methodology. But we're really at a kind of crossroads with really exciting opportunities now in generally in biological research, but particularly in nutritional research, where we can generate big data of high quality from new technologies such as digital devices, mobile clinical devices, remote clinical testing, so testing that can be done at home, for things such as your genetics or your microbiome, so the bugs that are in your food. We can also capitalise on people's interest in finding out more about how healthy they are and their health outcomes by taking part in this home testing and sharing their data back in this citizen science approach that Nathan mentioned in his introduction. And as well as these new technologies allowing us this wider reach, these new technologies are also giving us an enormous amount of data from just a few tests. So for example, from a single metagenomic reading, so this is a measurement of the bugs in our poo, this gives us thousands of data points. But once we've got all this high quality data, what can we do for it and with it, and how can we make it of value for us in nutritional science? And this is what we're really capitalising on in this PREDICT programme of research, which, as I said, is this collaboration between academic institutions, including King's, MGH in Boston, Stanford University, Harvard, Tufts, and ZOE, the startup tech company. And this programme of research involves a number of very large-scale nutritional trials, which I've summarised here and have been conducted since 2018. And these have been conducted at a really unprecedented scale, but also pace. So the speed at which we've done this work has been quite phenomenal compared to, our traditional research trials that we do in nutrition. And this is allowing us to advance our knowledge from one study to the next in a time frame that we just haven't been able to do in the past. And I've summarised the studies here. Due to time, I'm not going to go through each of these studies, but there will be access to these slides after, so you can have a look at these and see what's complete and what's going on after. What I want to focus on for about the next 10 minutes before I finish up talking a little bit about our COVID symptomat is our PREDICT-1 trial. So the PREDICT-1 study aimed to predict individuals' metabolic responses to food using a very integrated approach. So looking at genetic, metabolic, metagenomic, so this is these bugs in our poo, and also meal context information, so about how we eat, what time of day it is, how much sleep we've had, etc. And the aim was to address three fundamental questions. So how much variability there is between people, what explains these differences, and actually can we predict these responses using machine learning? And what makes our work so special is that, like I said, we're looking at these integrated responses, but also the interrelated multi-directional pathways that we often see in biological and nutritional research. And this just hasn't been done before in nutritional research because huge amounts of data is required to unravel this complexity, like I mentioned earlier, of these many determinants, us as individuals, but also of the food. And this just wasn't possible from our traditional small-scale randomized controlled trials that generally have looked at single factors. Also, what we're doing that's a little bit different to some of the big nutritional studies that have been done before is that we're looking at what we call postprandial responses, and I'll explain a little bit about what I mean by this. So traditionally, health responses to food and nutrients and generally clinically have been measured in the fasting state. So many of you will be told to go to the doctor fasting if they want to look at, for example, your glucose or your cholesterol levels. But we actually consume multiple meals throughout the day that contain mixed nutrients of fat, carbohydrate, and protein. And a typical response to a mixed meal is shown here, where there's a short, sharp rise in glucose, which is this blue line, which peaks around 30 minutes and returns to baseline about 2 hours. And there's a prolonged elevation in blood fat after a meal from the fat in the meal, which we call triglycerides, which is this green line. And this returns to baseline around 8 hours postprandially. And this combined metabolic response, so this increase in glucose and this increase in blood fat triglycerides, is what we call this postprandial response. And if we map these postprandial responses onto a typical eating pattern where we consume multiple meals, so we'll have maybe breakfast, lunch, and dinner, and 2 to 3 snacks a day, you can see that we actually spend most of the day, so about 18 hours, in this state of metabolic flux, in this postprandial state. And these metabolic fluxes in glucose and triglycerides influence health outcomes such as cardiometabolic disease, type 2 diabetes, and other associated kind of chronic diseases. So given that we spend most of our day in this postprandial state, and given that it has a really strong link to disease, it's really important that we look at these postprandial responses to food and that we try and move away from focusing just on the fasting effects or the long-term effects of diets. And it's these postprandial responses to food that actually underpin most of our chronic effects of diets on health as well. And this is exactly what we're doing in the PREDICT-1 study. So I'm going to just spend a minute outlining what we've done in the PREDICT-1 study, so how we've run the clinical trial. Again, I'm aware that many of you are non-biological or nutritional experts, but I think it's important I just spend a minute going through what we did so that you can understand in context the results that I'll spend the following slides presenting. So the PREDICT-1 study was what we call a multiple test meal challenge study. It was in 1,100 healthy volunteers. We had 1,000 in the UK, which was our main cohort, and we had 100 in the US, which was our validation cohort. And our study consisted of a clinic day and then a 2-week at-home phase. And the study design that I'm going to show you here is very similar to all of our subsequent PREDICT studies. So during our really tightly controlled clinic day, volunteers attended our unit at St. Thomas's Hospital, fasted, so they had nothing to eat or drink since the night before. They had a series of fasting measures for body composition, so weight, how much fat there was in their body. They had blood collected to measure genetics. They had a stool sample taken to look at metagenomics, so the microbiome or the bugs in their poo, and blood samples for a whole host of different biological assays. They then consumed a test meal, which was in the form of a very specially designed muffin that was super high in fat and carbohydrate, and then they had multiple blood samples taken over the next 4 hours. After 4 hours, they had another test meal, again a muffin, and then there were more blood collections made up until about 6 hours. And participants then started a 2-week at-home phase, and this was done entirely remotely, which again was a really novel aspect of this study, where we really capitalized on novel technologies. And during this 2-week at-home phase, they consumed multiple test meals, again in the form of muffins after an overnight fast, which varied in macronutrient composition. So it varied in the amount of carbohydrate, protein, fat, and fiber. And these were done in duplicate as well. And I'll come on to show you why we did it in duplicate in a few slides. And during this 2-week at-home phase, we measured sleep and exercise from activity monitors that our participants wore. We continually monitored their glucose levels from continuous glucose monitors. We measured their triglycerides and their insulin secretion from dry blood spots, which is a relatively new technology as well. And we also monitored hunger and mood using our ZOE app. And the dietary assessment, which is basically the measure of what people are consuming, so what foods they're eating, which is actually really challenging in nutritional research. We developed a whole new app for this, and we developed a real-time app and a dashboard. And this integrated weighed food records, barcoding, and also pictures. So people would take a picture of what they were eating in real time, they would scan the barcode, and they would also weigh everything, weigh it whilst they're cooking it, post-cooking, and also any leftovers. And we had live nutritional support from a whole team of nutritionists who reviewed everything that was logged in real time on their dashboard. So this enabled us to collect detailed and really high quality dietary data at a really huge scale that hasn't been done before in nutritional research. So our participants in the UK were mainly recruited through the Twins UK cohort. So we had about 65% who were identical and non-identical twins. And this is really important and a real plus of our study, because it enabled us to really explore the genetic contributions to how we respond to food. So we've had 1,100 healthy volunteers complete the study. Just to give you an idea of the scale, we had 32,000 of our lovely muffins consumed. We had 132,000 meals logged by the participants. We recorded 2 million continuous glucose monitor readings, and we recorded and measured 28,000 triglycerides. This is our blood fat measurements. And this is a really huge amount of detailed data compared to what's typically done in nutritional research. And this density of really big data collection has given us the opportunity to look at responses to food at a scale that just hasn't been done before, using these really standardized and very tightly controlled approach, but also at multiple time points, and also using multiple different meals of different nutrient composition. So this has created for us, just from PREDICT-1, and not even considering all the additional data we have from our other PREDICT studies, already the largest and the most detailed database of responses to food, from which we can really start to unravel determinants for the variabilities in responses. And the scale of this variability is really strikingly shown here. And this is our results in blood triglycerides. So remember the fat in our blood, and also in glucose. And this was published 2 weeks ago in Nature Medicine, which for me as a nutritionist is really exciting, because it's quite rare that nutritional science gets published in these very high impact clinical journals. And what you can see here is the really immense variation in both triglycerides and glucose in response, remember, to identical meals, and also in apparently healthy individuals where you'd expect to see less variation. The mean and the median is shown as the blue and the red lines that are down here. And you can see that this mean is really in the median, just not that meaningful at an individual level. So again, like I showed you earlier, how meaningful is this red and blue line to the person up here and the person down there? Same goes for glucose, and we find a similar amount of variability in all of our other outcomes, whether it's inflammatory mediators, insulin, and also in our microbiome measures as well. So we've seen this huge variability between individuals, but what about the variability within individuals? Now, for machine learning and precision nutrition to be effective, it's really important to determine how much of this variability that we're seeing here is independent of what you would get within an individual with repeat measures due to confounding factors or day-to-day variability. And because, as I mentioned earlier, our meals are given in duplicate, we've been able to look at the variability, not just between individuals, but also within individuals. And this is plotted here with the response to one meal plotted against its duplicate. So these are meals with identical nutrient composition. And what you can see is that the variability within an individual, shown by the coefficient of variations below, is considerably smaller than it is between individuals, which is really promising for personalised nutrition. And what we've been doing is starting to unravel what's causing these differences in responses, so causing the differences between individuals, but also trying to look at what's causing some of the noise or the variability within an individual. And one of the real strengths of this study is the twin population that I mentioned earlier, that about 65% of our participants are twins. And what this does is it enables us to estimate the genetic contribution to postprandial responses. And we find that there's a huge variability in responses even between identical twins. And this is shown here from the response of one identical twin plotted against the response of another identical twin. Now, if all responses were determined mainly by genetics, the dots should all sit on a straight line. And we see this for height, as you'd expect, because height is mainly determined by genetics. But you can see for TAG and for glucose just how much the dots are deviating from the straight line, suggesting that it's not all about our genes. What we can also do, because again we have both identical and non-identical twins, we can quantify this using what we call ACE heritability modelling. And using this kind of modelling, we found that 5% of the triglycerides and about 30% of the glucose response is due to genetics. So again, this shows us that a large proportion is modifiable and not down to our genes. And I think this is a really important finding, and it's really exciting because it shows to us that lifestyle interventions really do have the potential to produce pronounced effects on metabolic health, and it's not all predetermined in our genes. And I think for a child growing up in the 70s, where genetic research was taking off, this is super exciting because I grew up at a time where we were told, oh, well, how your body is decided by your genes. But this really shows it's not the case. I think what it also illustrates to us is that genetic tests on the market claiming to tell you how you respond to foods or the best diets for your genes are really only giving you a tiny, tiny piece of the puzzle. And personally, based on our research, I think a little bit of a waste of money. So we've got all this high resolution data sets, as well as looking at the contribution of genetics. What we can do is we can start to tease apart the other factors that are determining these postprandial responses in this integrated way that I mentioned before. And this, again, like I said, is unlike previous work that's just focused on single determinants. So what we've been able to do is quantify the relative contribution to both the triglycerides, glucose, and also other outcomes. But we've just got two triglycerides and glucose shown here. And what's most interesting to me, certainly as a nutritionist to highlight, is that the nutrient composition of the meal, which is shown in the dark purple here for triglycerides and here for glucose, only actually explains a small amount of the overall variability that we see for both triglycerides and glucose. And yet what we see is how we eat, which is what we call the meal context, explains, certainly for glucose, a lot more. So the time of day that we eat, how much sleep we had the night before, how much exercise we had, the timing of the exercise, for example, is really important. And again, I think this is super exciting because it shows to us that we can control our own responses to food, and that there really is huge scope for personalised nutrition. But again, it also emphasises the need to look at multiple factors when offering personalised advice. So we have all of this great data. We've looked at determinants, but where do we go from here? Well, we've then utilised machine learning to do more sophisticated analysis to predict individuals' responses to food. And we can predict an individual's glucose response, for example, to a given food with about 70%, 77% accuracy. And this is shown here from our measured down here versus our predicted results. And we're refining this further and further with all of our additional predict studies by getting more data to feed into the model. And these additional predict studies are also enabling us to address additional research questions, which the data show up to be of importance in, like I mentioned at the beginning, in a really high pace way. So we're currently probing the data from Predict1 and our other studies to look in more in depth about how the individual differences in the main determinants that I just showed you in the spider diagram, how they influence these postprandial responses, as well as how these differences in postprandial responses influence these subsequent health outcomes. And the biggest challenge really now is what can we do with this data, and how can we capitalise on these new technologies to translate to simple and actionable advice at an individual level. And some companies are already offering personalised nutrition advice, but generally, as I mentioned earlier, normally they're only looking at one single input, such as, or single outputs, such as genetics or microbiome only. And like you saw from our spider diagrams, our research shows it's crucial to look at the many integrated determinants. But we also need to think about what are the prospects for personalisation at population level, and how do we go about, once we've got all of this information, actually affecting change in people. And there's been a fantastic programme of research carried out by the EU called the Food for Me project that has looked exactly like this, that how effective is personalised nutrition? Does it actually implement change, and what's the best way to go about this? And I think really the main finding to highlight from that is that they do find that personalised advice does outperform standard one-size-fits-all advice on many different nutrition-related health outcomes. I think we also need to be mindful when we're thinking about personalisation is that there's many factors outside our control. So in the case of nutrition, factors such as food availability, convenience, and cost. And also, I think what's really is that we should, something's popped up on my screen, that we should remember that we can personalise and advise as much as we want, but ultimately we should ensure that we don't take away the enjoyment from food, that food and meal time should all be pleasurable experiences. So where does this leave ZOE, and where does this leave our research? Well, ZOE's created a personalised ZOE scores, and this combines machine learning algorithms from our Predict1 study, plus a whole breadth of expert scientific expert input, and detailed information about individuals to predict the foods in the diet that is best for each individual. And they're launching a product next month, which is involving a nutrition food logging app, so similar to what we were using in our studies, and a testing kit that tests many different blood measures as well as the microbiome. And after completing the food logging in the app and the testing kit, individuals will receive personalised insight reports on their results and also a personalised action plan. And this will guide people on the best foods and meals to eat for their unique metabolism. And it's supported by a whole team of nutritionists so that we can ensure advice is easily implementable for individuals, but also ensuring that we maintain the pleasure of the food. And there's a lot more information on the science and the ZOE experience on their website, which is joinzoe.com. And for us as nutritional scientists, this collaboration has really significantly advanced the relatively new field of personalised nutrition, and there's many exciting academic projects and publications that are arising from this. So this fantastic partnership between academia and a startup has really resulted not only in advancing nutritional research, but it's also resulted in an actual tangible outcome of a personalised nutrition experience, which is underpinned by this robust science. So I want to just finish very quickly with 2 slides to illustrate how this partnership has produced other benefits beyond nutrition. And this is the COVID symptom study app. As a quick bit of background, the week before the UK went into lockdown, Professor Tim Spector, who's the lead scientist on the PREDICT programme and head of the Department of Twins Research and Genetic Epidemiology at King's College London, and Jonathan Wolfe, one of the CEOs at ZOE, came up with the idea that it would be really good to use the ZOE engineering and data expertise and the King's College London epidemiological expertise to develop an app to track COVID symptoms and to try and understand what symptoms are predicted of COVID, as well as how prevalent they are, the diseases in the UK. So on the 19th of March, which was the week before lockdown, they had the idea. And on the 24th of March, only 5 days later, so the Tuesday of lockdown, the app launched. And by the 25th of March, so only within 1 day, we had 1 million users. And we now, really excitingly, we have 4 million users, which is really phenomenal. And this app has allowed us to map and predict COVID hotspots and people's likelihood of having COVID. And it's highly valuable given, firstly, all the issues that we had very early on with testing, and secondly, the ease and accessibility of an app versus driving hundreds of miles for a swab test, which we know is only about 70% accurate in itself. So how does it work? Well, it's a free app. Users of the app are asked to log their symptoms every day. And if they happen to also have a test, they can also log if they tested positive or negative. And this has allowed us to predict COVID based on clusters of symptoms, and most importantly, symptoms which originally were just not part of the government recognised symptoms. And the top 3 are fatigue, cough, and loss of smell and taste. And based on our prediction algorithms, we produce daily regional maps of estimates of symptomatic COVID, which you can see here. And because we have this huge database of COVID symptoms and users, we're now expanding this to look at what determines the likelihood of getting COVID and its severity. So we're starting to collect more data around demographics, phenotype, and diet and lifestyle. And we're using a similar approach as we did in PREDICT, where we can unravel what determines the severity and chances of catching it. And although very fortunately we've got a huge decline in cases, it still has ongoing value to support whether it's with contact tracing or expedite a safe lockdown, but really importantly to detect a second wave if unfortunately that does occur, but very quickly, region by region. If you want to find out more, again, there's daily updates on the COVID website at covid.joinzoe.com. There's blogs and all details on the scientific publications around this work. So I hope what I've shown you is what's really achievable by combining very traditional academia with AI within a startup company, and how it can really transform and really press the fast forward on not just nutrition research, but biological research. It's been a huge effort to undertake PREDICT, and many of those that were involved in PREDICT were also involved in the COVID symptom app. And so particular acknowledgement Zoe and Jonathan and George at Zoe, who have been phenomenal in their vision, and also Professor Tim Spector, who heads up all of the science around this. And thank you very much for your time listening to me. Okay, any questions?
Thanks so much, Sarah. Yeah, we've got a couple of questions. First one maybe is from Robin Allenson, who's an entrepreneur based in the Netherlands. Do you think the variability caused by age comes primarily from metabolic damage? And similarly, does the impact of meal timing being reduced the older you get have to do with poor sleep or accumulated metabolic damage?
So that's a good question. So we see that there's many interacting factors. So time of day, for example, becomes very important, is very important when you're younger. Yet once you get above 60 years old, the time of day that you eat just isn't important. And so as well as looking at a personalised level, we are starting to look at in a stratified way so we can answer those sort of questions. And so as you get older, your metabolic control is more impaired. And so a lot of the factors that might be more pronounced in younger people, such as sleep, such as time of day, appear to be less pronounced in older people, actually.
Cool, gotcha. Then we have a question from Andreas Fragner, who's in finance at a company called Jetstone in London. And he asks, in the Predict1 study, was there a particular motivation for splitting the training and validation cohorts along country lines between like the UK and the US?
No, obviously, as part of the machine learning process, we did have holdout data from the UK, and the US was our true validation data. It's always advantageous, particularly in nutritional research, to look at different populations, to look if it holds in a different population where there might be different dietary patterns. It adds strength to what we're doing. There was also a practical reason that we started the US study significantly later, well, about 9 months later, or 7 months later, sorry, than the UK study. So we were already utilising the UK data.
Okay. We got maybe one last question from William, who's in London, and he asked, what criteria beyond glucose response accuracy do you think is needed for a machine learning system to directly contribute to personalizing nutrition? Like, would we have to predict other things in addition to glucose response?
Nathan, I'm sorry, my audio was very poor then. Do you mind repeating the question?
Yeah, sure, no worries. The question is, do we have to predict more than glucose response using machine learning for an ML system to actually contribute to personalizing nutrition advice?
So if I understand, the question is to say, is it important to look beyond just glucose when looking
correct.
at? Yeah, definitely, and that's something we're doing. I've used glucose to try and keep it focused, because it's also one of our primary outcomes. We also believe that the change in blood glucose and blood fat is what has subsequent effects on the many other outcomes, such as inflammatory mediators, such as hemostatic and lipoprotein factors and oxidative stress. So it's the glucose and tag postprandially that impact a lot of the downstream effects. But certainly for the ZOE scores that we're using as part of the product, we're using a very integrated approach, looking at glucose, but also a whole host of other factors that we build in.